Digestive system part 1. Points to be covered in this topic: introduction, parts of digestive system, anatomy and functions of accessory glands. Let's see the introduction. The digestive system consists of the gastrointestinal tract, alimentary canal and its glands. The functions of gastrointestinal tract are ingestion, digestion and absorption of food and excretion of waste products. Parts of digestive system is divided into 8 parts: mouth, pharynx, esophagus, stomach, small intestine, large intestine, rectum, anus. First one, mouth. It is also known as buccal cavity. It is upper expanded portion which forms from the beginning of the alimentary canal. It is divided into 2 parts. First one is vestibule. An outer part, it lies between the lips and the inner lining of the cheeks externally and gums and the teeth internally. Second one, cavity of mouth. An inner part, it is bounded by teeth and mastoid bone at both the sides, palate above and below the tongue. The important structures of mouth are tongue, teeth and salivary glands. First one, tongue. It lies in the floor of the mouth and it is attached to the hyoid bone. Teeth. Teeth are concerned with mastication depending on the age at which they rise. Teeth can be classified into 2 types: A. Permanent teeth, B. Temporary teeth. Third one, salivary glands. There are 3 pairs of salivary glands. There are parotoid gland, submandibular gland, sublingual gland. Parotoid gland, they are the largest salivary glands and located just in front of the ears. Stenson's duct is a duct in each gland which opens on the inner side of the cheek opposite to the second upper molar tooth. Submandibular glands, they are the smaller than the parotoid glands. Each gland lies under the angle of jaw. They have duct called Wharton duct and it opens near the midline under the tongue. C. Sublingual glands, they are the smallest salivary glands and lies under the tongue. They have several small openings through which they pour onto the secretion directly. Functions of salivary glands. First one, cleanses the mouth due to the bacterial action of lysosome and IgA, immunoglobulin A, one of the immune system antibody. Second one, creates a feeling of oral comfort by lubricating action. Third one, dissolve food chemicals so that they can stimulate the tongue's taste. Fourth one, helps to form a bolus, a ball of food, by the action of mucin, thus facilitating swallowing. Second part, pharynx. It lies between the mouth and the esophagus. It consists of 3 parts. Nasopharynx. It lies behind the nasal cavity and extends from the base of the skull to the palate. Next part, oropharynx. It lies behind the mouth and extends between soft palate above and upper opening of the larynx below. Third one, laryngopharynx. It is the lowest part and lies behind the larynx. Third one, esophagus. It is a muscular tube which extends between the pharynx and cardiac orifice. Pharynx and cardiac orifice of stomach below. It lies between the trachea in front of and vertebral column at the back. Esophagus contains sphincters at its upper and lower ends. These sphincters relax during swallowing. Fourth part, stomach and its digestive function. Stomach is a dilated portion of the alimentary canal. It receives the food from the esophagus. It lies in the upper part of the abdominal cavity below the left half diaphragm. Structure of stomach. It consists of 4 coats. First one, peritoneal coat made up of serous covering. Second one, muscular coat made up of longitudinal, circular and oblique fibers. Third one, submucous coat made up of alveolar tissue. Fourth one, mucous coat made of mucous membrane. Secretions of stomach. Glands of mucous membrane of stomach continuously secretes gastric juice. The secretion of gastric juice due to a reflex mechanism through vagus nerve. Gastric, a hormone secreted by the action of foodstuffs on gastric mucous membrane. Psychological effects produced by taste or smell of the food. Gastric juice contains 5 types of enzymes. First one, pepsin. It is an enzyme produced by the glands present in fundus and body of stomach. In the presence of hydrochloric acid, pepsin converts into protein into peptone. Second one, renin. It is an enzyme which curds milk. It involves the conversion of caseinogen, the soluble protein of milk, into the insoluble casein. Third one, hydrochloric acid. It is secreted by paratile cells of gastric glands. Its concentration is about 0.4% of gastric juice. The chloride is derived from the sodium chloride in the blood. Now the hydrogen ion combines with the chloride ion to form hydrochloric acid. Functions of hydrochloric acid are neutralization of saliva and acidification of food, helping the action of pepsin in converting to the peptone. Antiseptic action by killing bacteria. Fourth one, intrinsic factor. It is a content of gastric juice which is necessary for the absorption of vitamin B12. This vitamin is necessary for the development of red blood cells. Fifth one, chyme. It is the product of digested food in the stomach. It is a semi-liquid form and it is passed on to the duodenum. Next, functions of stomach. There are 5 types of functions. First one, mechanical function, digestive function, protective function, homeopathic function, excretory function. First one, mechanical function, is divided into 2 types, storage function and formation of chyme. A storage function, food it is stored in the stomach for a long period of time and then emptied up into the intestines slowly. This provides enough time for proper digestion and absorption of food in small intestine. B. Formation of chyme. Peristaltic movements of stomach mix the bolus with gastric juice and convert it into the semi-solid material known as chyme. Second one, digestive function. Gastric juice acts mainly on proteins. Proteolytic enzymes of the gastric juice are pepsin and renin. Gastric juice also contains some other enzymes like gastric lipase, gelatin lipase, urease and gastric amylase. Third one, protective function. First one, mucus protects the stomach wall from irritation or mechanical injury by virtue of its highest viscosity. Second one, prevents the digestive action of pepsin on the wall of the stomach, particularly gastric mucosa. Fourth one, homeopathic function. Intrinsic factor of calcium secreted by paratile cells of gastric glands plays an important role in erythropoiesis. It is necessary for absorption of B12 from GI tract to the blood. Fifth one, excretory function. Many substances like toxins, alkaloids and metals excreted through the gastric juice. Small intestine. It is the part of the alimentary canal which extends from the pyloric end of stomach to the cecum. First part of large intestine. 3 parts of small intestine. First one is duodenum, shortest and wider part. Second part is duodenum, thicker and vascular. Third part is ileum. It is thinner than the jejunum and less vascular. Digestion in small intestine. Acidic chyme from stomach enters into the duodenum. Then it mixes with the alkaline intestinal juice called succus entericis. Alkaline secretions from the liver and pancreas. Enzymes responsible for the digestion: enterokinesis, epicin, sucrase, maltose and lactose. Enterokinase, it converts a trypsinogen of pancreatic juice into the trypsin. Epicin, it converts polypeptides into amino acids. Sucrase, maltose and lactase, it converts the corresponding disaccharides into the monosaccharide. Absorption in the small intestine. The absorption of the digested food occurs in small intestine through villi. They are minute projections present in the inner mucous coat of the intestine. It gives the velvety appearance to the intestinal mucous membrane. Each villus has a central core composed of one artery and one vein, a strand of muscle. A centrally located lymphatic capillary is lacteal, connective tissue that adds to the support to the structures. Large intestine. Large intestine colon extends from the end of the ileum to rectum. Large intestine consists of the following parts: appendix, ascending colon, transverse colon, sigmoid colon, caecum. Functions of large intestine. There are four functions: digestion, absorption, secretion, excretion. Digestion. It is carried out by microorganisms of colon. They act on undigested and unabsorbed residue from the small intestine. Absorption. After all the small intestine, only water and glucose are absorbed in the colon. Third one, secretion. Mucin is only secretion which lubricates the colon, facilitates the passage of fecal matter. Excretion. Iron and some purgatives are excreted in the large intestine. Seventh one, rectum. It occupies the lower posterior part of the pelvis, extends from the sigmoid colon and anus. The lower part of the rectum is dilated and it is called rectal ampulla. Next one, anus. It is a small canal measuring about 1 inch in length. The opening of anus is guarded by the sphincter called anal sphincter. This sphincter is under voluntary control. Anatomy and functions of accessory glands. There are 3 accessory organs that aid process of digestion which are located outside the alimentary canal. They are liver, bile ducts, gallbladder, pancreas. First one, liver. Liver is the largest abdominal organ. It lies in the right upper quadrant of abdominal cavity, inferior to the diaphragm. Anatomy. It contains 2 lobes, right lobe and left lobe. They have 4 surfaces: superior surface, inferior surface, anterior surface, posterior surface. Microscopic structure. The liver is made up of small hexagonal units present in large number called lobules. Each lobule has a central vein or intralobular vein surrounded by several portal triodes. The portal triodes consist of portal vein, bile duct, hepatic artery. Blood supply. Blood is brought to the liver by hepatic artery, portal vein, hepatic veins. Hepatic artery, it supplies oxygenated blood to the liver. It is a branch of coeliac plexus which arises from the abdominal aorta. Second one, portal vein. It carries a veins which lie in between the lobules of liver. They subdivide and ultimately form central veins. Third one, hepatic veins. It carries the deoxygenated blood from the liver to the inferior vena cava. The central veins of each lobule join to the form sublobular veins. The sublobular veins unite to form several hepatic veins. The hepatic veins joins with inferior vena cava. Functions of the liver. There are 8 types of functions of liver. First one, secretion of bile, synthesis and storage of glycogen, formation of urea by deamination of amino acid, synthesis of plasma proteins like albumin and globulin, storage of vitamins and minerals, synthesis of heparin, natural anticoagulants. 8th one, detoxification and purification of blood. In the above diagram, there are functions of liver. Detoxification involves drugs and alcohol, fatty acids, steroid hormones, ammonia, urea, environmental toxin, allergens. Metabolism. Conversion of T4 to T3. Detoxification of fat. Next, immune system. It contains viruses and pathogens. Maintenance of hepatic and portal vein immune system. Next, protein synthesis, blood clotting, formation of prothrombin, cholesterol transport, lipoproteins, immune function, formation of globulins, oncotic pressure, formation of albumin, copper bioavailability, ceruloplasmin, Storage of micronutrients like minerals, copper, zinc, magnesium, iron, vitamin A, D, E, K, B12, blood sugar balance, storage of glycogen, production of bile needed for the digestion, GI antimicrobial. Next, bile ducts. The secretion of liver bile is carried through bile ducts, which are formed by the union of biliary caniculi. They present in between the lobules of liver. The bile ducts formed from the right and the left lobes of liver unite to form common hepatic duct. Later, the common bile duct unites with the pancreatic duct in the duodenum at papilla called ampulla of water. Third one, gallbladder. It is a pear-shaped hollow structure. It is located on the inferior surface of liver. It is divided into three regions: fundus, body and neck. Layers of gallbladder. There are three layers: serosa, muscularis, mucosa. Serosa is the outer covering. Muscularis, middle layer, helps in the gallbladder to contract and squirt its bile into the bile duct. Mucosa, the inner layer of folded in duct that allow the gallbladder to expand. Duct of gallbladder. The duct through which gallbladder opens is called cystic duct. It joins with the hepatic duct to form the common bile duct. The common bile duct then joins with the pancreatic duct, which opens into the duodenum. Functions of gallbladder. Reservoir of bile. Bile is an alkaline fluid produced by hepatocytes in liver and stored in gallbladder. It is a mixture of water, bile salts, bile pigments, electrolytes, cholesterol, phospholipids and triglycerides. Liver produces 500 ml to 1000 ml of bile per day, but the capacity of gallbladder is only 30 ml, so it is concentrated in gallbladder. Second one, bile salts. Bile salts present in the bile are sodium taurocholate and sodium glycocholate. They act as emulsifying agent. It absorbs digested lipids. Third one, bile pigments. Two important bile pigments are bilirubin, biliverdin. They are waste products produced from the hemoglobin. Fourth one, pancreas. The pancreas is located behind the stomach in the upper left abdomen and surrounded by other organs. It consists of head, body, tail. Head. It is a wider part of pancreas. It is positioned toward the center of abdomen. The central section of pancreas is called the neck or body. The thin end is called the tail and extends to the left side. Structure of pancreas. Substance of pancreas contains a number of lobules of secretory cells called acini. In between the acini, there are groups of endocrine cells called islets of Langerhans. The small ducts emerges from these lobules. These ducts unite and reunite to form pancreatic duct of Versung. The duct begins at the tail and emerges from the head of pancreas. It enters the duodenum along with the common bile duct. Secretion of pancreas. There are two types of secretions: exocrine and endocrine secretion. Exocrine secretion, pancreatic juice with digestive in function. It is transferred to duodenum through the pancreatic duct. Pancreatic juice containing the following digestive enzyme: lipase, amylase, trypsin. Lipase, it converts fats into fatty acids and glycerol. Amylase, it converts starch into maltose. Trypsin, it converts peptones into the amino acids. Endocrine secretion, it is secreted by islets of Langerhans. It is directly poured into the circulation. This secretion contains two different hormones are secreted by two different cells of islets of Langerhans. First one, glucagon. Second one, insulin. Glucagon is secreted by alpha cell. Insulin secreted by beta cells. For more details, subscribe to our YouTube channel Pharma Pro EDU. Please like, share, comment.